Thanks everyone who was able to join today. I'm really excited to introduce today's speaker, Dr. Christine Cutucache, who's the Associate Professor of Biology and Haddix Community Chair of Science at the University of Nebraska, Omaha. Dr. Cutucache received her Bachelor's of Science from the University of Nebraska and her Ph.D. in Genetics, Cell Biology, and Anatomy from the University of Nebraska Medical Center. Her research lab primarily studies tumor immunology. However, she's also active in science education research, currently a member of the National Council on Undergraduate Research. She works to design new teaching methods to improve learning retention. She's also the founder of the Nebraska-wide STEM for You K-8 outreach program that focuses on problem-based learning activities outside of the classroom. Today, she'll be sharing her insights on how we can use science communication to advance our initiatives both inside and outside of the lab. So with that, Christine, would you like to take it away?
Thanks so much, Kokila. I appreciate the nice warm welcome and a very special thanks to you, Melissa, and to Rosie for getting us set up today and letting me share the message with y'all. All right, with that, I will share screen. And hopefully we're good. Can you see fine, Kokila?
Looks good.
Your screen.
Awesome. Thank you. You know, it's always a little tough with the webinar format. The pandemic has certainly posed some challenges for us, but, you know, instead of a full day of travel and, you know, the enjoyment of nice warm weather, maybe a nice cool breeze, I am instead joining you, of course, as Kokila mentioned, from Nebraska. It's rainy. We have 55-mile-an-hour wind warning. So just a little juxtaposition between both California and Florida. So glad to join you all today. As Kokila mentioned, my name is Christine Cutucache. Most of my students refer to me as Dr. C because I have one of those hard to pronounce last names. And certainly I've had the journey of being a wet bench researcher all the way through being a discipline-based education researcher. So I've been able to see firsthand and experience firsthand the need to be able to talk to both scientists and non-scientists, and even in the middle, kind of switching science areas. So I'd like to share with you a few stories today, but of course, none of these stories would be possible without our wonderful funders. So before I move on, I'd really like to just express my gratitude on behalf of the team for the great support that we've received, not only just in funding, of course, we certainly appreciate that like everyone else, but I think every time we receive a grant or a contract from an agency, it really shows that they have faith in us and they are counting on us to do the hard work, and we don't take that lightly. So thanks so much to our funders. With that, I'd like to share a few stories, one being about some of my experience as a wet bench biomedical researcher, because I think that folks can relate, particularly from your group, most specifically to that story. Then I'd like to pivot and share a bit about my work in discipline-based education research. You can also think of that as science education research. And then we can talk a little bit more about SciComm more generally and some tips and tricks for your toolkit today. So for the first story, let's start with one of my favorite molecules in the world, caveolin-1. If we weren't on webinar format, I would be asking, how many people have heard of CAV1? Who else has it as a favorite gene? Raise your hand. But caveolin-1 is so interesting because if you were learning about science maybe 20 or 30 years ago, it was only involved with cell membrane. But now we have a much better understanding of it in that it really is this juxtaposed role. We refer to it often and fondly as a Janus-based molecule because it serves as both a tumor suppressor and an oncogene, which goes against anything you've ever heard about in a cancer biology course. But the interesting part of it is that the function is really based on location within the body. So that's how it can go between those two. Caveolin-1 is also responsible for TCR-induced actin polymerization. That's often what we hear about it with. Same with membrane rafts and connections between cells. Others have described it as the orchestrator of the immune response in lymphocytes. And I'd like to tell you a little bit more about that story. Short version of that story. So many malfunctions in lymphocytes have been associated with immune deficiency or challenges with the immune response. And one of the types of malignancies that I've worked on, chronic lymphocytic leukemia, is the most common adult leukemia. And one of the challenges that we see across the different types of CLL is that there is immune dysregulation. So some other groups have done some great work, like John Bird's group, finding an immunomodulator, for example, to help repair that issue. But our team has been really interested in identifying what molecules are upregulated that are contributing to immunosuppression that we might try to identify treatments to manage. But we've also been really interested in mechanism, right? So it's one thing to see what's up, knock it down, that's your treatment. But it's a whole other thing to identify the mechanism of action of CAV1, for example, in chronic lymphocytic leukemia. And we also have studied T-cell leukemias and lymphomas for the very same. And one of the things that we saw about 10 years ago is that there's probably a role of CAV1 with immune synapse formation. And so we set up a series of experiments to test this very thing. One is that we suggested that if we saw a misregulation in CAV1, then immune synapse may not be forming. There may be immunosuppression as a result of that. So we ran some in vitro experiments where we knocked down CAV1, starting in an antigen presenting cell, and then looked to see if there was any change in immune synapse formation. Immune synapse is detected just by filamentous actin staining between the T cell, the smaller lymphocyte, and the B cell in this example. So if we don't knock down CAV1, we have a lovely immune synapse. When we do knock down CAV1 in the antigen presenting cell, we don't see the synapse. And then we can rescue this phenotype by using a scrambled knockdown. We ran the very same set of experiments with the flip side, so knocking CAV1 down on the T cell side and not manipulating the antigen presenting cell, and saw the exact same thing. So these data really validated what we saw in our gene expression profile data. And so we then translated that into a mouse model. So if we knock down CAV1, we could make heterozygotes and see changes in chronic lymphocytic leukemia progression over time based on how much CAV1 there was. And you can see by gross histology of the spleens at the bottom right that there were substantial changes. But if you look at this slide more broadly, and this is the take-home message, you have everything from in vitro tissue culture work to flow cytometry, and then mirroring translation and then process of those tissues. We were doing the very same with human or clinical samples as well. So the important part is that we had folks who are biomedical scientists and clinicians working side by side, which is very representative, of course, for the many teams that you work in. And the conversation about science is relatively easy when sharing these types of data, right? I mean, the folks working on flow understand pretty well the in vitro experiments that help to justify running the flow experiments, so on and so forth. But then as we started to take this mechanism and learn more about it, we start to look at things like cell-to-cell communication, more about the synapse formation, movement of cells, and now we're looking at pathways. And so now you add in the biochemists and the geneticists, but we're still basically talking the same language, right? If we start to run simulations now to identify whether there's a change as a result of CAV1 being upregulated or downregulated in either an immunocompetent environment or an immunocompromised environment, now we have even new types of data that we can look for. So for example, we'd have to include folks like immunologists and network scientists, because certainly we could run these experiments in vitro, but it's much more expedited to run it in silico. And if you're a curmudgeon about some of the in silico data like me, I always like to have the in vitro and the in vivo data to support and check the in silico data, and that's exactly what we did. So we needed folks like network scientists to work alongside us biomedical researchers to be able to translate these types of things into the in silico environment and then run those assays. And then given that, we could turn up or down any of these molecules and see the change in either an immunocompetent or immunocompromised situation, which gave us really impactful information in short order. It was also the first time that a T cell was modeled in this way. So we continued on with this approach. Certainly we took information from gene expression profiles and then ran it in silico and worked with then also computational biologists and engineers to then build out more of these types of in silico models. I mentioned I'm a curmudgeon, right? So we get the data from the in silico. Can we validate it using either in vitro or in vivo or both models? And we were able to do exactly that, which is a wonderful result. It's a wonderful outcome. It was super exciting because I'm showing you just snippets of a few different studies today, again, because our theme is SciComm. Our theme is not chronic lymphocytic leukemia or splenic marginal zone lymphoma or T-cell lymphoma, but the idea being that it's multiple teams working together in order to move these things forward. So we translated these back to in vivo, and ultimately, fast forward a bit, we submitted a provisional patent on these very data. So super exciting, but working with lots of different teams. On the T cell side, this was translated then also looking for prognostic and diagnostic markers. So now we're working with bioinformaticians to run these data, make these massive scale models to then run additional predictions to inform how we ask questions in vitro or in vivo. So that's the short story about a cancer biology example of all of the different teams that have to come together in order to advance a research initiative. Certainly I shared with you a bit about going from the in vitro to the then in vivo and my in silico using the models and predictions by working with computational biologists and engineers to build those model systems, and then ultimately translating back to an in vivo environment. We did generate a model of the C4 positive T cell, and we could determine that the predictions that we made actually translated, which is always exciting when that happens. We know there are a lot of setbacks in science, and so when you get those wins, you have to celebrate them, of course. And then we identified some other upregulated and downregulated molecules that ultimately then moved forward in the lab for additional testing and then ultimately application. So we moved ahead with particularly splenic marginal zone lymphoma is what we've spent the most time on most recently. But how could any of this actually be successful? Well, it's really due to collaboration. And certainly, you're all experiencing that science is based on collaboration. We must collaborate with others. We get the opportunity to collaborate with others in order to make our science even better. And, that's not the case, even 1980s, 1990s, if you, look at a lot of the papers that were published then, a lot of times it was sole author or just, 3 authors or less. And now you see the Nature publications with hundreds of authors. So the science becomes better thanks to those really effective collaborations. The SciComm is pretty easy, right? You're communicating and code switching doesn't happen yet, right? You're working with folks that already understand the data, already understand the application. Maybe as we got to the in silico part, that got a little bit farther than the expertise that we had on the team, but we're still talking scientist to scientist. So the communication was really pretty easy, right? So let's take another example. In the case of discipline-based education research or science education, I'm really interested in learning how students learn best. And I'm also really interested in particular interventions that lead to the retention of students. I would love to see 100% of students that enter into an undergraduate, to pursue an undergraduate degree to graduate. But we know that that doesn't happen, unfortunately. And then add on the challenges that we face in STEM in terms of retaining students, and then even more so retaining folks within the profession. So my group's also been studying how we can best support students to keep them in the pipeline. And one such intervention includes reaching out to younger children and getting them excited about science early. So we work in an after-school time space where we serve students once or twice a week throughout the entire academic year, and then in most cases all the way through the summer as well, to provide enrichment around STEM. So think about it as the way that students can get their hands dirty with STEM. It's hands-on, minds-on. And so it's a really exciting, fun after-school activity for kids. But I really care about how we can best support our undergraduate students. And so, of course, our students are the instructors in the class, basically. And so we support them through that through 3 different ways: teaching, research, and mentorship. Certainly, they're offering teaching for the kids, and we help them learn to complete active learning strategies. They go through a variety of training, and we give them the resources needed to be most effective in that classroom. We also have students who participate in research as part of this. So they can collect information such as doing pre-post testing, interview structures, focus groups, et cetera, to get more information about the outcomes related to our program. And then lastly, we have mentorship, and that's through a variety of different strategies. So for example, we have faculty mentoring undergraduates. That's perhaps the most obvious one, but the undergraduates are also then serving as mentors to the kids in the project. A third mode of mentorship is our more senior undergraduates also mentor our more junior undergraduates. So they get mentorship from 3 different capacities. And so we study the effect of these 3 mechanisms within the program. So certainly we're working with kids, we're working with undergraduates, faculty are actively involved, but of course this is a research project. So how do I share the information that's gleaned from this intervention with others? Well, one of my stakeholder groups is instructors. If this is really an intervention that's of value to undergraduates, then other undergraduate faculty should help support undergraduates with finding this opportunity. And then administrators, right? We're always having the need to justify best practices. I mean, that's best practice in and of itself. So how do we share the data in a useful format? Because even though as scientists, we love to write articles, we love to read articles, not everyone shares that same thing. So the way that I have to communicate some of this information to others takes a variety of formats, everything from like a short technical report or a policy paper to a presentation as opposed to a journal article. On the discipline-based education research side, we collect data in different ways than I showed you on the biomedical side, right? I'm not running flow cytometry, I'm not doing any tissue culture. In this instance, we're using mixed methods research, which, you know, as a biomedical trained scientist, it took me a while to not see it as measles, mumps, and rubella. So if that's how you first saw my acronym, please know I'm right there with you. But we use mixed methods research, which includes a compilation of quantitative data, qualitative data, and then triangulating those data as the third portion of it. And so we collect a variety of types of data, and then we analyze those, of course, and share out the results with others in the field, discipline-based education research experts and other pedagogical experts. Certainly that takes the form of a journal article most predominantly. And so we try to reach the audiences that need to get the information, which in some cases is outside of what we were typically used to on the biomedical side, of course. So CBE Life Sciences education is probably the closest one we have to the traditional sciences that I was trained in, for example. But we have to also reach folks who are going to be able to understand the information and translate it within their context. So other faculty at institutions, graduate students, postdocs who are interested in having or contributing to an outcome that supports undergraduates with the added benefit of supporting kids. So it's certainly important to reach each audience where they are and make it relevant to the audience. And I've certainly seen the value of having both quantitative and qualitative data and then bringing those two together in order to tell a story. In most cases, it's a much more compelling story. And as one of my colleagues puts it, you know, we can share graphs all day, but I've never seen someone cry from seeing a graph. If you take folks around and maybe look at some artwork or, you know, take them through a different experience for sharing information, you can really motivate folks in different ways. So the way we share information has to vary by who the audience is. Certainly in the first example I shared, it's basically biomedical researcher to biomedical researcher. In this instance, you're not having to justify the work, right? You're also not having to give a lot of information about the methodology used or why you should or shouldn't use those types of methods. The question and answer portion is pretty straightforward. However, you know, we kind of go up a level, so to speak, when then we submit our work for publication, because now our peers are reviewing our work. And so certainly it's still going to be folks with similar expertise, but they may pick apart things a little bit more so than just someone else on the team who's also a researcher in that area. We jump tremendously and enter the SciComm territory when we move outside of sharing only with scientists, right? So we can share our data. That's not scientific communication, it's communication, right? But as we went to file our patent, for example, we met with a lot of attorneys, and the attorneys would ask questions like, what is this? Why is this? Why would anyone care? And it was a whole new frame of reference about how to communicate our work. So when we're talking with the non-scientists, we really have to think about how can we demonstrate impact to a wider community? And then of course, the multiple million dollar question is how do we effectively lobby for things like earmarks and appropriations? We really have to identify what motivates the individual or individuals we're trying to reach. And then after we understand what motivates them, launch a campaign for cascading messages that make it very impactful, understandable, relevant. And being relevant can be really challenging because the world changes so fast. And certainly the news cycle that we're on, social media, things come so quickly. And so how do we capture the attention of our audience? And that's important, right? Because I have yet to meet a researcher or educator who does not need funding for something. So we certainly need to be skilled salesmen and women, so to speak, to address these needs. And let's say, you know, you don't need funding for anything. Great. You still have to share out that information. And again, how are you going to connect with that audience and make it most relevant? So science has turned out to be a little bit different than I expected. I really expected that it would look more picture on the left-hand side of the screen where, you know, I've got my earbuds in, I'm listening to my music, pipetting away, building graphs, running a variety of experiments, happy, right? But what I have really learned is that science is more this. And the picture on the right shows someone writing, but it's really meant to demonstrate communication. We have to communicate at every stage. And it's actually to our advantage to communicate at every stage. When I had my first federal grant, I didn't know all of the things that would come due at the annual report time, right? But perhaps like other folks, I waited until, you know, the notice came that the report would be due and you could preview some of the questions. And it took me a substantial amount of time to prepare that report because I didn't know what was going to be asked. And I hadn't saved evidence of support along the way. So much, what I'm trying to say is that the more you can communicate your science and in multiple ways and over time, the better. So test hypotheses, certainly do the fun and engaging science, but then also communicate the impact. And that communication of the impact really has to be packaged for a variety of audiences to be effective. When we think about this collaborative process of science, it really is this circle, right? It's a group of folks working together, but we all rely on data from other people, right? Everyone, when they join a new research group or start an experiment, they're relying on things like literature reviews or maybe even raw data that was handed over from a past lab mate. And they're using that information to start asking questions. And certainly every aspect of science now is really done in teams. And so we have teams then working dynamically together to move the initiative forward. Science happens thanks to funding from a range of resources, certainly, but we still need funding in order to make it work. And that funding really happens from, say, federal or state appropriations, maybe the traditional federal agencies that we typically work with as scientists, or it could be, providing evidence that the team can do what's proposed, meaning in the proposal that goes to those agencies. So certainly you have to share why your science is relevant, why it matters, but also share that you can do the work, you can accomplish the work. Remember, as I thanked our wonderful donors and funders at the beginning, they had faith in us to get the work done, and we shouldn't take that lightly. And of course, teams get funded. So it's really important to have the right folks on the team and the right folks in the right seats on the team. I was looking for some great pictures for this presentation. And as I Googled my lab, I was able to see so many of my outstanding students over the years. So I just did screenshots of the ones that showed up. And then, of course, I've added additional photos of some of my outstanding colleagues that I get to work with on projects now, too. And the take-home message from all of these is that teamwork is really what leads to effective SciComm, because effective SciComm comes from effective communication. Effective communication comes from preparing and practicing the very things that we need to be effective communicators, such as teaching in the classroom or teaching in the after-school environment, or sharing information on a regular basis and getting feedback about that information. Did it land? No. Why didn't it? Oh, I need to make it more relevant for my audience, for example. In case you're not convinced yet, this is the holdout slide. This is for all the holdouts out there. So certainly folks have become more and more passionate about effective SciComm, but now we're to the point where our federal agencies are requiring us to be effective at scientific communication. If we don't, they will not fund us. So we want to make sure that we can, of course, effectively use taxpayer dollars and contribute to scientific literacy. We would love for everyone to have an understanding of science and to even be really excited about science. But certainly there's this need to improve scientific communication. There's some distrust of scientists. There's distrust that we see, in the news each and every day. And so we really need to be thoughtful as researchers in conveying a message that is going to be heard and supported as others hear it. So for example, the National Science Foundation has a very specific procedure for including things like SciComm, and that falls into what they call broader impact. How will you make your work be translated well outside the lab? That's broader impact. And we know that if a message is not conveyed in an effective way, it can steer people away from STEM. And we certainly don't want that to happen. I would like everybody to be a scientist. Just kidding. But science is so cool. Everyone should have a love and appreciation for science, perhaps. So this is how that happens, broader impact. Certainly intellectual merit is the other major criterion that NSF uses. And so those go hand in hand. But it's really evident the proposals that give great thought to broader impacts and make it highly translatable. The same applies for the other major agencies. So for example, HHMI, Howard Hughes, They even have a newer instrument, the biointeractive. And so that is a resource, a SciComm resource that you can access today, HHMI's biointeractive. Certainly, this is the group that has created the translatable work such as the CFAGs and engaging undergrads and community members more broadly. So very specific initiatives that are trying to support and encourage SciComm. The proposals that already have that embedded are a little bit more favorably supported because they have a plan, of course. And then NIH certainly focuses on disease treatment, disease prevention, but they also really focus on economic growth and expanding the workforce. And that's through preparing the next generation of biomedical researchers. That's another way of saying broader impact, but it's through translating this work again through SciComm that we can really make impact on scientific literacy, especially. This is one of my favorite quotes. It has hung on the wall in my office for a very long time. I recently moved offices, and so I need to print out a new one of these. But this is from John Holdren, who was the past science advisor to President Obama. And he says, "I've said for a long time that every scientist and every engineer in this country should tithe 10% of her or his time to engage with public policy and with public education on science and technology issues. We no longer have the luxury of staying in our laboratories, of sitting at our desks, working on advancing our science and engineering disciplines. We have to interact with broader society in ways that communicates what we're doing, why we're doing it, and why it matters. The scientific community has to get better at telling informative stories about how science works and about what it is doing in support of the aspirations of the American people." If this is not a call to action, I don't know what is, right? 10% of our time, we can certainly do that to make sure that we can contribute to science literacy, and we can do that through scientific communication. And the nice part about SciComm is that you can match your own interests and abilities with the message. More on that in a minute. How can you reach folks then with your science communication? Start with the why, just like we just heard. Why should the audience care? How might they be impacted? Maybe it's a positive impact, maybe it's a negative impact. What would the inaction lead to? How would it impact their lives? What's the time frame for the action? If we don't move forward on X, then Y will happen by Z. We also really need to think about how we frame the data we need to share. So for example, if I say, well, the prevalence of disease A is 44.5%, to others that may sound quite boring. It may not reach them. But if instead I say 1 in 2 people, ... makes it a lot more understandable. We can also really encourage the participation and expand reach by including a call to action. By getting the audience involved, giving them a sense of urgency, that's another way to really better reach the non-scientists. So as an example, hopefully folks have heard of Be the Match, the bone marrow donation registry. So you might say, I've talked about lymphoma today, and before you leave today, I would really appreciate you signing up to be a member on the Be the Match registry. Sign up before the seminar is over, right? You're telling them what to do, why to do it, what the impact is, and then a time with which to do it. So I mentioned the biointeractive as a resource, and I think other resources are immediately accessible and other resources might need a little bit of planning and time. And so for example, I shared a bit about our work in reaching kids who are in kindergarten through 8th grade, but also our work with undergraduates. And so scheduling meetings with the folks that you're interested in reaching will give you a lot of information about what they're motivated by, what is or isn't relevant to them. As I've learned, middle schoolers are brutally honest. So if you'd like feedback about any number of things, go share it with some middle schoolers and they will give you some great feedback. You can also subscribe to SciComm focused things such as the Alan Alda Center as well. And that will allow you to access a range of resources, see examples of SciComm in action, etc. A lot of the national meetings have SciComm embedded within them at this time. So the network of STEM education centers often has resources, as does AAAS. So you could go, as part of a conference you're already attending, go and learn more about SciComm that way. Or you could just offer to dig in, right? Maybe you really want to get more experience, and so you're going to offer to run the social media page for your lab, or you're going to prepare a technical report on topic A. Maybe you're going to go and speak to a non-scientist group about your work. Some great options include like your local Rotary or Chamber of Commerce, maybe your alma mater, right? So just going and speaking with folks that are outside of your research area and sharing a bit about your work. So I hope you've enjoyed a bit about the stories that I've shared, with everything from cancer biology and the importance of translating that work with a variety of audiences, everything from biomedical researchers through patent attorneys, and of course with our funders and reviewers there, through our science education work, and then ultimately thinking about how we might approach SciComm to make it most effective moving forward. So if you're ever looking to connect, I hope that you'll add me on LinkedIn. Really happy to visit with you there or get you connected to other resources. But at this time, very much a thanks to Kokila, and we can welcome questions.
Hey, thank you so much, Christine. That was a fabulous talk. You touched upon this really briefly at the end, but could you maybe elaborate a little bit more on your thoughts on just how valuable social media is as that form of communication? I know, science Twitter is a really big thing now, and especially when talking to both academics and non-academics, it seems to be a pretty equalizing platform. So maybe you could share a little bit more on your experiences there.
Certainly, thank you. So, 4 or 5 years ago, I was the holdout. I didn't see the importance of social media. I tried to avoid social media. But as you can see on the slide before you, I'm asking you to connect with me on social media, right? So I've come full circle. The reason I came full circle actually and dove into the social media was because on one of our NSF grants, we had our external reviewer come, and he was like, "Wow, your project is great. Everything's going so well. Your students are well supported. The research is cruising along." And I was waiting for it, right? Like, what's the downside? What's coming? What have we messed up on? And he said, "But you're not on social media, so you're not meeting students where they are." And so that resonated with me, right? If anyone tells me I'm not meeting my students' needs, okay, I heard it. Let's make some action. And so I hopped right on social media from that forward. And the second most important thing I heard from another colleague more recently was when I was trying to think about how to set up our communications more broadly on a number of things. I said, well, I think that here's plan A. And my colleague was like, that's your brand, that's your reputation, that's how you're communicating out. You may want to think through this a little bit. And so both of those colleagues were spot on, right? Because obviously we have a lot of researchers indeed on the social media platforms, Twitter and LinkedIn, for example. But we also have non-scientists that want to get information, and they want to know whether something is reliable or not. And so really it's our duty as scientists to share on social media so that folks can understand what is reality and what is not. Having a scientific eye review something from science and then posting it onward sends a different message than someone who may not have the same amount of experience. And so I think it is our duty to really contribute on things like social media platforms. And so, yes, I think that's where the field has gone, rightfully so. It took me, a little bit to embrace that, but it was great advice for me. So I think it's a wonderful way to move forward. Thank you for the question.
Yeah,
Other
thank you.
questions?
Hi, Christine. I really liked your talk. I had a question following up with what Copila was saying, which is that like during COVID, obviously, there's a lot of information running about whether true or not on social media. And I'm wondering about how you see social media sometimes, especially on Twitter, as more like a sound bite, which can be taken out of context and like misinterpreted. And especially with scientists, I know a lot of us, or maybe historically, hedge on saying like absolutely people need to get vaccines or absolutely this needs to happen because there's always, you know, a 0.01% chance that it could be bad or something. And I think scientists tend to hedge on that. And so when you get these sound bites, sometimes I think they can go in the wrong way and like make people think that, you know, something is happening that isn't. And I'm wondering like, how do you think about drawing the line between, you know, being succinct, but also covering your bases as a scientist to be accurate?
Yeah, great question. I think that most scientists try to hedge their bets with including links to more information. So certainly you provide an abbreviated, short, succinct version, but then a link to more. That doesn't mean folks will go and look and read the additional information, but it's almost like the asterisk, right, that comes with it. And so that's a really nice way to do it. If you haven't looked at the HHMI's biointeractive resources, they do very much that kind of a setup, you know, here's a short snippet, click here for more. And so I think that's one way to do it, but yeah, it's really hard. And even with that, I mean, you know, I don't know how many journal articles I've read in my lifetime. I probably don't want to know how many journal articles I've read in my lifetime. But I think it's even the same going between different disciplines of science because there is so much in that context. There's so much in the historical context. One of the diseases we've studied in my group, splenic marginal zone lymphoma, is almost a nice fresh start to study because it wasn't identified that long ago. So in that example, you can actually read chronologically everything that has been done in splenic marginal zone lymphoma. That is few and far between, right? And of course, leukemias, lymphomas predate that, you know, obviously, but at least it's something that we can understand a little bit more comprehensively. But then if you say, well, cancer, it's a huge category of things, you know, and things may vary between types, even like massive buckets of cancer, such as solid tumors versus hematological malignancies. And so it's a slippery slope. It's hard, right? But I think that a lot of folks that are non-scientists are express some frustration that science is not always translated outside of the quote unquote ivory tower, right? So sharing something that may not be as complete, but links to more information, I think is better than not sharing anything. Thanks for the question.
Yes, thank you.
I'm not sure if anybody who's joining us, an attendee, has questions. Feel free to either type them in the Q&A or you may be able to raise your hand and we can probably call on you and unmute you at this point too. So feel free to do that.
If not, I do have another question.
Go for it.
Full of questions. So Scripps, I'm sure like a lot of institutes, you know, is trying to push for more science communication and getting their trainees access to, say, practice material or opportunities to practice. And I'm wondering when, you know, each, at least at Scripps, I'm not as familiar with other places, when you work for a PI, they're your direct boss. And so they get to decide what you do. And I'm wondering if you work for someone or maybe at an institute that doesn't really support, say, 10% of your time or even 1% of your time going towards these efforts, how would you suggest people either push back on the institute level or just finding opportunities themselves? Because it sounded like some of the options, like social media, obviously we can all jump in on, but some of these opportunities to like go to a high school or something like that are easier if there's already an established program.
Yes, certainly. So I always like to have data to negotiate. So I would encourage you to bring data. Part of those data would include the like 2021 employer surveys. So if you Google, you know, the employer surveys, I think it was NCES that put that one out. And some of the top things that employers are requiring of the workforce today are communication skills, collaboration skills, critical thinking skills, right? And SciComm accomplishes a lot of that training. Certainly you can get some of that by being at the bench, problem solving, of course, right? I mean, I remember at times we would put money underneath the thermocycler of like, please work, right? So there are things you can suggest perhaps to an advisor, a mentor of, I'm really considering a career in SciComm. I would appreciate more opportunity to learn and practice SciComm. That doesn't always work, right? And so the next negotiation may be, here's what employers are saying they want and need. I'd like to make sure I'm competitive on the job market. That may not land, right? And so I've heard it all. And so instead, then you could engage in SciComm more on the weekends or anytime where you don't have to be in lab very specifically, so that you could do a presentation at the Rotary. They meet at 7 p.m. and maybe you could time your experiment so the next check is 10 p.m. instead of 7 p.m. or something like that. But it's a challenge. And it's a really aggressive job market right now. I know that a lot of folks are walking right into jobs that are wonderful. That's great. I graduated during a depression, basically, housing crises, et cetera. And so the market is so different now than what it was, most certainly. But the thing is, even if folks can get right into a job today, we want to make sure that they'll be well supported to advance and elevate in their role, right? Because one of the main reasons that people leave an employer is they don't see the growth opportunity, or they don't feel like they're being supported in a way that is going to encourage them to learn more. And so that's the other part of the negotiation piece, right? Is sure, I'm going to be trained and prepared for job A, but how can I be promoted in job A? And ironically, I just did a kind of it was a panel, but I think of panels always as like a fireside chat, right? You have the folks sitting at the front of the room with the microphone, the questions come and you're just kind of discussing them back and forth. So I just did one of these with graduate students in another institution. And certainly folks can gain the entry-level skills to get the first job, but the things that promote folks are communication, communication, and communication. And communication can take a range of descriptors, but one that we shared is conflict resolution. It is really important to be able to figure out how to mitigate a conflict, how to approach it calmly, how to make all sides happy at the end of the day, or at least agree on something at the end of the day. And gosh, talking with non-scientists sounds like a great way to learn those types of things to help you later on. So I hear you. I know it's a challenge always. We have so many different demands on our time. And, I imagine the mantra has not changed too much from when I was in graduate school of publish or perish. And so certainly we want to contribute to our publications, but you can do publications in SciComm as well, right? And it's more so important, I think, to make sure we can talk to federal agencies or appropriations of any kind and get things supported, because the trickle down effect, right, is then, okay, those policymakers get it, but then they're also sharing with their stakeholders and constituents more about science. So it gets everybody talking about science more. So it's really a win-win-win. Yeah, thanks for the question. Great one.
Thank you.
Thanks. I actually have one more quick question that I was thinking about that I'd like to get your insight on. So at a place like Scripps, we don't really have undergraduates. We do some outreach. I know with COVID kind of things changed a little bit, but we have done a lot of K through 12 outreach in the past. But for the most part, our primary students on campus are our graduate students. So from your experience doing, your discipline-based education research, are there any tips or tools you may have for the faculty that teach graduate classes, or our graduate student TAs that are usually the senior graduate students TAing the incoming students, that maybe there are some tools or tips that would help enhance sort of communication at that level.
I'll follow your question with a question. What are comps like for you at Scripps? Do you write a grant?
We basically write like our dissertation proposal, and that's our main exam for our candidacy.
Okay. So a lot of schools address that very need with having comps that are actually a grant writing exercise. And so that's what I would suggest. That doesn't mean that folks have to write a full grant. I'm sure that lots of folks are writing F31s, F32s anyway, but something like putting together the specific aims page and some justification and maybe budget also, things that are going to be highly translatable. I think those are really good opportunities to understand the importance of messaging. And so then you could say, okay, great, I can put this together, but I'm not sharing it with anybody. Is that really a value added? Well, I would encourage you then to take that kind of product and indeed share it with other people. I'm sure there's a lab right down the hall that maybe studies something different that you could share that information, get some really active feedback on. In one of my grad classes, I actually require that students write an NSF IUs as part of the class. And I think that initially it's super intimidating. It's very laborious, it's very challenging, but it's a huge value added because then they have the practice and how are they going to justify it? And then they get to see a mock review of their proposal. So they can't say anything like, oh, what I meant was we would have N of 65, not N of 55. They have to just sit there and watch and listen to the mock review. And so they can see whether what they put to paper was understandable. Was it easy to follow? Was it well organized? Did it make sense given the literature base? Is it going to be translatable, right? So I think there are exercises that you can do even in a short 1 hour time frame where, yeah, if you prepare, that specific aims page, pick it apart amongst the different groups. And that can be one of those kind of forays into that. So yeah, writing grants, super fun, but I think it's a good exercise.
That's really great. And I think you really hit the nail on the head. I mean, you can do all you want, but if you don't share it with other people and actually get the feedback and the experience, it's not really as relevant of an exercise. So that's awesome.
And getting that feedback also helps, again, with the communication and conflict resolution, right? I remember still my first paper, and I don't even think it was rejected. I think I just read the reviews as just being awful, right? Like, I must be a horrible scientist if they're saying to make all these changes. No, they were giving me really useful information to take and improve my paper. And it was improved thanks to their great advice. And so it gives us more opportunity to practice getting comfortable, hearing negative feedback, giving negative feedback. It's all constructive, right? But there's something about our brains like, oh, they're saying something negative about me. No, they're saying something constructive about the grant, right? So I think it's a really worthwhile exercise, definitely.
Yeah, thank you. Melissa, any final questions before we wrap up?
I think I wrapped up my questions, but thanks.
Yeah, well, thank you so much again, Christine, for taking the time to speak with us today and give us all your valuable information. Thanks to everyone, our attendees who were able to join. I hope this was a useful seminar for you all. We will have this recording available along with the recordings from our previous seminars on our YouTube channel, so keep an eye out for those links. And hope you can join us for our next event on the 29th with Dr. Eric Topol. So thanks, everyone.
Thank you. Thanks for the invite. Take care, everyone.